light, phase contrast, electron microscope, phase contrast electron microscope. Next one, also structure and function of ultra structure and function of plant cell and organelle. This was the cell organelle mainly uh, chloroplasts, mitochondria. What are the single already discussed that of mitochondria, chloroplast, different types of cell. And next one, single membrane models are the and next one is the black blue. Next
Sir,
one is
voice is
the
not clear, sir.
okay. Okay. Now it's clear.
Yes, sir. SPS, sir.
Okay. Cell organism, single membrane models, double membrane models, different types of the cell organisms also we discussed. And the chromosomes, especially chromatin plus DNA equal to chromatin, DNA plus chromatin equal to chromatin. Next one, the different types of the chromosomes, the size and shape. Okay, shape especially the structure, the centromere based on the chromatids. The two chromatids are the attached by the centromere, primary centromere, the secondary centromere. Okay, and next one is the shape of the chromosome: V shape, I shape, L shape, I shape, different types of special type of the chromosomes also carbonic chromosome, Pauldine chromosome. Next one is okay. Today we are discussing the uh, cell cycle and apoptosis. And next one is the DNA also. Okay. Cell cycle mainly
Okay, sir. Cell cycle and
apoptosis and DNA also. Okay. Okay. Cell cycle. Uh, what is the mean of the cell cycle? Cell cycle is present in all living organisms. Have only living organism. Uh, they cannot see in the non-living organism, especially they can see in the living organism. The cell division cycle, most of cells is the consist of four coordinate process. What are the mainly these are the cell division cycle? The most of the cells is the contain the four coordinated process. What are the coordinated process? Cell growth, DNA replication, and distribution of the duplication of chromosomes to daughter cells. And next one is the cell division. These are the these are the four. Uh, okay, these are the four is uh, the four is the main important. The coordinated, the cells cycle is the coordinated. There are the four steps: cell growth, DNA replication, and next one distribution, duplication, chromosome to daughter cell, and cell division. Okay. The cell cycle is also a sequence of the cell growth and division. That the both are the cell growth and next one division also the sequence of the cell growth and division. Mainly these are the time takes complete cell one cycle is the generation time. The time it takes to the complete one cell cycle is the generation. What are the generation time? Approximately 24 hours of the generation time. The cell cycle The totally in the cell cycle they are containing the G1 phase, G2 phase, S phase, and next one M phase, meta phase. Okay, all our steps are involved in cell cycle. Okay, mainly they are containing the four steps: DNA replication, cell growth, and next one cell division, and distribution of the duplication chromosome to daughter cells. Okay, next one. A cell cycle is the process from the end of cell division to the start of the next cell division. Right? The cell cycle is the process from the end of the cell division to the start of the next cell division. Okay, it's also process of from the end of cell division to the next start of the next cell division. Most of the eukaryote cells, approximately 24 hours is the cell cycle. Already we discussed 24 hours. Okay. Most of the eukaryotes already we discussed the cell cycle only they can see in the some of the most of the living things. Living things. Okay, living things. Eukaryote cells is the divide approximately every 24 hours. The cell cycle is developed to basic two parts. One is the interphase and next one is the mitosis. Interphase and mitosis. Okay. Phases of the growth is the already mitosis and interphase. Interphase they can divide the three spaces is there. Okay. G1 phase is also called as the primary growth phase. Synthesis, mainly formation of the DNA replicate. The mainly is the primary growth phase, G1 phase. S is the synthesis, is also called the DNA replicate phase. The G2 is the secondary growth of phase. These are three stages are the collectively they called interphase. Interphase. Already we discussed the cell cycle is the two phases. One is the interphase and the two parts of the cell cycle, interphase and mitosis. The next one, the interface are that they can divide the G1 phase, S phase, G2 phase. Okay, and the mitosis also, mitosis, next one, mitosis and cytokines. These are the cell cycle. Okay, this is the diagram of the cell cycle. These are the G1, S phase, and G, G, these are the G1, S phase, and G2 is the interface. These are the only, these are the mitosis M phase is also called as the M phase. These are the diagrammatic of the cell cycle. Okay, these are the diagrammatic cell cycle. Okay, these are also diagrammatic, especially in G1 phase. The G1 phase is the cells are the mature conditions. G1 phase already G1 phase and S phase means synthesis phase. DNA replicated phase. DNA replicated phase. DNA replicated phase. And next one is the cell prepare for the digestion. The G2 phase. G2 phase. And the next one is the mitosis, especially M phase, prophase. Next one metaphase. Next one anaphase, telophase, and the finally cytokinesis. These are the cell division. Mainly, these are the identical into the identical cells of the identical cells. Okay. Most of the some of the somatic cells are the multicellular organism. Most of these somatic cells are the multicellular organism. The adult human being approximately 1 lakh billion cells and the all originate from the single cell. The fertilized egg cell. Somatic division cells is undergo process of termed as the cell cycle. Cell cycle. Cell division is cell cycle and mainly somatic dividing cells undergo go to a process is termed as a cell cycle. Somatic dividing cells process termed as a cell cycle. The cell cycle is ordered to series of the event leading to cell replication, copies of the parent. And some of the multicellular organism, most multicellular organism, the most different cell, exitive and exitive and the cell cycle length. And the next one is the without the some of the cells are the exitive cells, exitive cell cycle. That the cell cycle is the primarily been fusion of the yeast, some especially Saccharomyces pro. Mainly these are the two strains, Saccharomyces, and the next one is the some of the mainly these are the strains are the Saccharomyces, Saccharomyces. Next one is the Pro. The both are the two strains have been especially useful for the isolation of the mutants and the block specific steps of the cell cycle. Okay, mainly the cell cycle is the three steps are the regular mainly is the blocked mutate blocked with the specific steps is the cycle exhibit the altered the regulation of the cell cycle. Especially three scientists of the Paul Nurse and the team Hunt discovered the molecular mechanism that regulate the various phases of the cell cycle. The mainly the Leland Hartwell, Paul Nurse, and team Hunt. The three scientists. The three are the Nobel laureates. Mainly the molecular mechanism regulate various phases of the cell cycle. Next one is the cell cycle interface. Already interface is the stage between the two division. It is the longest phase. Especially the interface is the stage between the two divisions. In between the two stages. What are the two? Especially it is the longest phase. There are also longest phase. Approximate interface is last from 89 hours. Already we discussed in the previous diagram G1 phase, S phase, and G2 phase. Okay. What are the characteristics of the interfaces? It is the resting phase of the cell. It is also called as the resting phase of the cell. Resting phase of the cell. It also is the longest phase. The interface is the longest phase. And next one is this phase is the prepare the cell for the mitosis. Mitosis. No division occurs into the interface. The metabolic activities are the high in these phases. These are the longest phase. Okay, there is no metabolic activities in the eye in this phase. What is in this phase? And interface. There are the longest phase, resting phase of the all cells. Next one is the no division occurs into the interface. The characteristics of interface. Next one is the G1 phase. Okay, G1 phase also we discuss the each and everything cell cycle. G1 phase mainly cell cycle. The cell cycle is ordered series the leading to the cell division. Essentially, cell cycle divided into the four. Already we discussed four major phase: G1, S, G2, and M phase. I already discussed the interface is the G1, S, G2 is the interface. Okay, G1 phase. G1 phase is the G1 is the gap period between the mitotic phase and the S phase of the subsequent cell. For example, G1 phase is the okay, these are the in between the say this one G1 phase in between the synthesis phase and the next one is the mitotic division is the G1 phase. The G1 phase also longest cell cycle phase. G1 is the typical is the longest cell cycle phase. Mainly is the G1 phase carry out the synthesis of the RNA and protein synthesis of okay, sir. Mainly the G1 phase is the gap period between the mitotic phase and the S phase of the subsequent cycle. It is the gap period. The first growth period is also called as G mean gap. G mean gap. It is the G. It is the gap period and first growth period. Generally, the stage loss the 25 to 50% of the total interface. Okay. During this phase, during the G1 phase, mRNA and the rRNA, tRNA, most of the RNAs are in protein synthesis inside. proteins and RNAs. What are the RNAs? Especially DNA and RNA, DNA, RNA. Next one is the especially RNA and protein synthesis. Different types of the RNAs, mRNA, rRNA, and tRNA. Okay. Next one is the mainly is the nucleus produced rRNA, mRNA and tRNA. These are the they can control the cell growth. Okay. This one is the G1 phase. These are the characteristics. These are the longest phase. And next one is the main important is that during this phase, the synthesis of the RNA and proteins. RNA and proteins. Okay, next one. Okay, next one is the S phase. S phase is also S means synthesis. During the S phase, DNA synthesis and the chromosome replication is initiated. In this phase is the mainly during the DNA synthesis, chromosome replication is initiated. S phase. DNA synthesis occurs, the DNA molecules, the duplicates. Synthesis means duplicate. The period is the approximately loss is the 35 to 40% of if interface. Mainly are the character DNA replication occurs. Histones are the synthesized. The histones covers each DNA strand. These are the characteristics of S phase. S means synthesis phase. S means synthesis. Mainly is the DNA replicate or the DNA synthesis. Mainly in this phase, DNA synthesis occurs. The DNA molecules are the duplicates. DNA molecules are duplicates. Okay, DNA molecules are in this phase mainly a copy of each chromosomes is formed. Already replicate, replication formation, replicate form. During this phase, the DNA synthesis occurs, the DNA molecules is the duplicates or the replicates. Mainly these are the replication of occurs. Histones are the synthesized. The histones are covered each DNA strands. Okay, these are the S phase, the characteristics of S phase. Next one, G2 phase. During the G2 is the next phase of the S phase. The mainly are the checks that the RNA replication is completed and prepare for cell division. The next, they can continue the cell division. G2 phase is the gap period between the S phase and the mitotic phase of cell cycle. S phase and mitotic phase of the cell cycle. G2 is also the secondary growth phase. What are the primary growth phase? G1 is the primary growth phase. G2, what are the importance? What are the characteristics of the G2 phase followed by the mitotic phase? Intensive cellular synthesis. Mitochondria and the chloroplast is divided. Energy stores in increase. Energy stores in increase. Okay. The next one is the already we discussed the interface is over. G1. What are the main important? This is the longest period. The G1 phase. Okay. G1 phase. Next one is the S phase. Mainly duplication of the DNA synthesis. And next one is that they can form the histone proteins also. Histone protein synthesis. Histone proteins mainly replicate. The histone proteins are covered by the histones are synthesized in S phase. The G2 phase is the secondary growth phase. Mainly is that the important is the synthesis of the intensive cellular synthesis. Energy stores are the increase in G2 phase. These are the interface. And next one is the mitotic phase are also called as the M phase. This division is the M stand for the mitosis. M mean mitosis. M phase. These are the divided into the 5 discrete phases: prophase, prometaphase, metaphase, anaphase, and telophase. Okay, prophase, prometaphase, metaphase, anaphase, and telophase. These are the 5 steps. There are the 5 steps is there in mitotic phase. Mainly in prophase is the chromosome condension. Mainly they can see in prophase phase, they can see the chromosome condension. And next one is the spindle fibers, especially the spindle fibers are the synthesize and resemble to the mitotic operators. And during the prometaphase is the nuclear envelope breakdown to small vesicles. Okay, especially in prophase are the chromosome condension. These are the important. And in prophase is the chromosome condension, and next one is the microtubules and the synthesized to the mitotic operators mainly during the prometaphase. Prometaphase mainly is the what are the important and break down the nuclear envelope into the form to the small vesicles. Okay, they can form to the small vesicles. Next one is the protein, especially protein structure and the kinetochore assembled to each centromere of the sister chromatids. Okay, sister chromatids. The breakdown of the nuclear envelope, the spindle fibers attached to the kinetochore as well as the arms of the chromosome. During the metaphase, especially in the first one is the prophase condension of the chromosome. Next one, envelope, nuclear envelope is breaking. They can form the vesicles. Okay, the vesicles are the attached to the kinetochores. In metaphase, especially, chromosomes cell is the equilibrium position midway between the poles is also called as the metaphase plate. Especially, they can form the metaphase plate. These are the metaphase plate, especially these are the made of the some of the magnesium ions and the calcium ions also. Magnesium ions and calcium ions also. They can form the metaphase. The third phase is the metaphase is over. And next one is the anaphase, especially in during the mitosis. Sister chromatids are the separated. Sister chromatids are separated. Okay, and finally the next one is the mainly the anaphase is the spindle microtubules toward the opposite poles. They can form the opposite poles. These are the anaphase. Okay, these are the anaphase. The spindle fibers are the they can separate the uh, polar regions. Okay, opposite poles, and they're the separated the spindle elongates. And next one, finally, the chromosome separation is complete mitotic spindles. The chromosome discondency during the telophase. Condensation, especially they can see in the in prophase and in telophase discondensation. The spindle fibers they can especially a group of condension and the telophase is the mainly they can form the discondensation. Especially in metaphase they can form the metaphase plate. Metaphase plate. Okay, metaphase plate. Okay, these are the five steps in mitotic division: the prophase, prometaphase, metaphase, anaphase, and telophase. Especially in anaphase, is the spindle pairs are they can convert the polar region, especially the two opposite poles. They can towards the opposite poles. Okay, especially these are the physical division of the cytoplasm, the term as the cytokinesis. Cytokinesis. When these are the physical division of the cytoplasm, the term as the cytokinesis. The process is the separated to two cells. When these are the two forms, they can form the two daughter cells, the identically chromosomes, the complaint. So these are the mitotic phase. In this phase is the approximately duration is the 45 minutes and to 1 hour. In division, mainly these are the in division in this phase, phase division of the nucleus, mainly the division of nucleus is also called as the karyokinesis. Karyo mean nucleus. Karyokinesis. Okay. Mainly these are the only occurrence in the eukaryotes. They cannot see in the in prokaryotes. Only they can see in the mitotic phase, especially in eukaryotes. Okay. Already we discussed the prophase, metaphase. Next one is the anaphase and the telophase. These are the already prophase. Will give the prophase, prometaphase. In prophase, especially they can form condension. Metaphase mainly metaphase they can form the middle plate. Mainly these are the metaphase plate. They can form the metaphase plate. Anaphase mainly anaphase they can form the sister chromatids. Okay, separated by the sister chromatids. The telophase decondension. Okay, these are the characters. M phase. Okay, these are the different mitotic prophase. Next one is the mainly prophase. Next one is the metaphase. Next one is the anaphase. Finally, form the telophase. Finally, they can form the two daughter cells in the mitosis stage of the mitosis. Okay, already cytokinesis means the physical division of the cytoplasm. The physical division of the cytoplasm is also called as cytokinesis. Mainly, cytokinesis is also the process of the cell division, which divide the cytoplasm of the parental cell into the two daughter cells. Okay, mainly cytoplasm, mainly the division of the cytoplasm. The term is also called as the cytokinesis. Cytokinesis. These are the process of the cell division. It occurs to with the two types of the nuclear division is also called as mitosis and meiosis. Mitosis and meiosis. Mainly these are the two types of the nuclear division. Okay, mitosis and meiosis. Okay, cytokinesis are the mainly two types of the nuclear division. One is the mitosis and the meiosis. Mainly these are the which occur in the animal cells and the plant cells also. The cytokinesis diagram of the cytokinesis. Next one is the mainly the plant cells are the membrane bound cell plates formed to the in metaphase, especially prophase. Next one is the prometaphase. Prometaphase, and next one is the metaphase. Metaphase, and next one is the anaphase and telophase. Okay. When in plant cells, membrane bound cell plate form to the in metaphase. These are the plates are the synthesized from the Golgi operator. These are the mainly Golgi operator. Golgi operators is the single membrane models. Golgi operators is mainly supplies the plasma membrane that separates the two daughter cells. Mainly synthesize the cell wall between the daughter cells and cell plate. The cell plate is the mainly actin filaments are formed into the perimeter of the cell. These are the mainly myosin also. Myosin and two tights with the belt, the cell pinched into the two daughter cells. Mainly they can form the cell plate. Okay, in cell plate. Okay, these are the cell division. Cell division. Next one is the how to control the cell cycle. Okay, regulation and the cell cycle is the binding. Next one is how to control the these are the mainly eukaryote organisms essential the cell cycle is the preciously coordinated. The mainly one phase is must be completed before the next phase can be begin. Proteins are the cytoplasm control the some are the mainly in this phase controlled by the some are the proteins. But mainly the network of protein kinases and phosphatase triggers. Protein kinases, and next one is the phosphatase triggers are the transition between the cell cycle. Mainly protein kinases are the involved in the cell cycle regulations. These are the very important. In CSR exam also they can ask into the some of the net set examination also. What are the protein? Mainly the cell cycle is controlled by the some of the protein kinases enzymes involved in the cell regulation. Protein kinases. Okay, protein kinases. Protein kinases. Okay, these are the next one is the cyclin dependent kinases also they can regulate the in cell cycle. Cyclin dependent kinase. Already we discussed the protein kinases involve the regulation of the cell cycle. These are the very important. Okay, next one is the cell cycle dependent the kinase is also called as the CDK. Cyclin dependent kinases is also controlled by the CDK enzymes. These are the serine tyrosine protein kinase. The term is also called as P34 CDC2. Mainly CDK is the serine tyrosine protein kinase. The term as a P34 CDC2. These are the mainly they can control the cell cycle. What are the protein? What are the protein? P34 CDC2 type protein. They can control the cell cycle, regulate the cell cycle. Mainly the protein kinase active enzymes, the regulate subunit is also called as the cyclin. Subunit is also called as cyclin. Mainly the cell cycle is controlled by the serine tyrosine protein. Mainly serine tyrosine protein also what are the P34 CDC2 in it. The subunit is also called as the cyclin. The cyclin attached, they can control the some of the cell cycle. Okay, cell cycle. The next one is the also cyclin dependent kinase also. Some of the especially the women have the more than 10 different protein kinases also available. This protein kinase essentially cell progression during the both G1 phase, S phase, and G2 phase, MM transition. Mainly cyclin, cyclin dependent kinase, they can control the cell cycle. That is very important. Mainly what type of the enzymes, what type of the proteins are involved in the protein kinase also. They can regulate the cell cycle by the protein kinase enzymes. Protein kinase, special kinase enzymes, especially protein kinase. Okay, these are the controlled by the cell regulation. Next one, cyclins also. These are the diverse group proteins with the similar to the core structure. These are the 16 different cyclins also have been identified in human being. 16 different cyclins are the identified in being. Tim Hunt is are the mainly discovered the in 1980s the proteins from the accumulate during the interface of the cell. And next one is the these are the destroyed during the mitosis. What are the mainly cyclins also? They gradually accumulate into the interface of the cell and the abruptly destroyed with during the mitosis. The cyclins also derived name by the sort cyclic accumulation and destruction. Cyclin, the mainly are the cyclins derived it's the name by this sort of cyclic accumulation and destruction. Accumulation and destruction. Accumulation and destruction. And the next one is the mechanism of cell cycle in the control. Already we discussed the what are the steps are involved in the mechanism of cell cycle controlled by the already mainly in some of the eukaryote it is the universal mechanism regulates the events of the entry to the G1 S phase, G2 and M phases. Mainly in G1 phases the steps are in the cell cycle is controlled. What types of the steps are involved in G1 phase especially? Mainly cyclin CDK complex are the express the first one. The cyclin mainly initiation start with the rising levels of the cyclin D in the cytoplasm. Mainly cyclin D binds the CDK4. Mainly is the binding with the CDK4. Mainly is the CDK4 enters the nucleus signals the cell prepare the chromosomes for the replication. Chromosomes for the replication. Mainly is the binding with the CDK4. Cyclins are the CDK4. Mainly is the G1 phase. These are steps are involved in G1 phase and the S phase also. Some of the cyclins E and A type are bound to CDK2. Mainly in G1 phase, the cyclins D are binds with the CDK4, and the next one is the cyclin E and A are type bound in the CDK2. Okay, the complex is also called as the SPF S-phase promoting factor. SPS means S-phase promoting factor. S-phase promoting factor. In S-phase also, DNA replication continues and the cyclic E is the destroyed. Already in S-phase, E and A type bounded in the CDK2 two protein, especially to CDK2, mainly in the end of the what are the main theme of the S-phase? The replication of the DNA, replicate or duplicate of the DNA. The finally cyclin is the E is the destroyed. Cyclin is the destroyed. G2 phase also cyclin B and A type B beings the rise up during this phase. M phase also is the mainly what are the steps are involved in the mainly is the control mechanism of cell cycle control G1 S phase G2 M phase assembly of the mitotic spindle. Next one is the breakdown of the nuclear envelope, the condension of the chromosome. All steps are involved in the cell cycle is the control. Mainly is the first DNA pre-replication complex assembly to the origin. Next one, G cyclin GDK activates is the CDH1. CDH1. Next one, G cyclin is the different steps are involved in the cyclin CDK activate expression of the S phase in the cyclin CDK components. And the G1 cyclin CDK phosphopyrolates and the S phase is the inhibitor. Next one is SCF. SCF means already we discussed the SCF means S phase promoting factor. S phase promoting factor also promoting degrade the phosphopyrolate S phase. Next one, cyclin CDK inhibitor. S phase cyclin CDK activated in the pre-replication complexes. Mitotic cyclin CDK activates early mitotic events. Next one is the APC CD20 protease degradation in the finally APC CD1 is the proteasome degradation mitotic cyclin. All are the steps are involved in the regulation. And next one is the next one cell cycle of the G1 phase, S phase, and G2 S phases also. Okay. Some are the regulation of the cell cycle in the budding yeast also. Is that the yeast is the belongs to the fungal member. The cycle of the Saccharomyces cerevisiae is regulated primarily point of the late G1 called is the start. Mainly is the passage through start is the controlled by the availability of the nutrients. Matting factor is also cell size. The yeast are dividing the budding forms from the just after start. Mainly is the regulating of the regulation of the cell cycle is the budding yeast. First one is the G1 phase, S phase, G3, and M phase also is the large phase. Approximate G1 is the largest phase. Okay, mainly the G1 phase is the start the nutrients, the mating factors, and the cell size also they can see in the cell cycle. Next one is S phase replication. Next one G2 is the G2 also is they can see in the finally they can form the mother cell too that they can form the daughter cell. The steps are involved in the cell cycle of the budding yeast. Okay, these are the cell cycle, some of the checkpoints. G1, G1 is the stop the DNA damaged and the unreplicated or DNA damaged in S phase. These are the some of the these are the checkpoints. There's also one checkpoint, and the next one is the checkpoint. These are the checkpoint. These are the checkpoint. Unreplicated or damaged DNA. Mainly is the damaged. Okay, damaged DNA is the one of the checkpoint. Mainly cell cycle is the what are the main conclusion of the cell cycle is the only they can see into the living organism. All cells are the reproduced by dividing to the two with each parental cell giving rise to the daughter cell in the completion of the each cell cycle cell division. These are the cell cycle. Okay, these are the cell cycle. Next one is the apoptosis. Okay. What is the meaning of the apoptosis mainly? 1 minute. Okay. Next topic is the programmed cell death is also called as the apoptosis. Okay. Program cell death, especially programmed cell is also apoptosis. What are the apoptosis? Is the natural occurring the process of by which cell is the directed to the programmed cell death. Programmed cell death. This name is given by the Greek name described in the following of the leaves. Apoptosis is the Greek name. Okay. The course apoptosis is accompanied by the characteristic changes in the cell morphology. Mainly is the genetically programmed cell death which leads to the tidy breakdown and the disposal of the cells. Morphologically, apoptosis is characterized mainly apoptosis characterized in the cell membrane with the formation of the small bulbs known as the apoptotic bodies, shrinking of the nucleus, chromatin condension, fragmentation of the DNA. All are they considered in apoptosis. Especially macrophages and the other phagocytic cells recognize the apoptotic cells. Mainly these are the apoptosis cell death. Okay, this leads to the cancer. Okay, this leads to cancer. Apoptotic cells and the remove of the phagocytes without the inflammatory phenomena is developing. Mainly these are the apoptosis diagram. Some of the characters are doing the apoptosis cell morphology. Okay. So these are the diagram of the apoptosis. Mainly these are the, for example, the viable cell. Next one is the apoptosis. The cell shrinks in the chromatin condension. See, this one is the cell shrinking and the chromatin condension. The next one is the form of the budding. Apoptotic bodies are the phagocyte is the no inflammation. Next one is, see, this one is the viable cells. The necrosis the cell swells. Next one the cell becomes the leaky, bleeding. Next one the cellular and the nuclear lysis causes the inflammation. Okay. What are the basic functions of the apoptosis? What are the basic functions of the apoptosis? Okay, these are the tissue homeostasis and development differentiation, the basic functions of the apoptosis. Next one is immunosystem, and next one is the mainly in 1 minute. Tissue homeostasis as the independently role of the development and differentiation process, especially in the embryo. Mainly, apoptosis is the some of the cells are the committed suicide through the well characterized intracellular program is also called as the programmed cell death is also called as apoptosis. Mainly, some of the adults are the responsible for balancing the cell population and the maintaining constant the cell numbers in the tissues undergo the cell turnover. For example, approximately 5 to 10 to the power of 11 blood cells are the eliminated in the daily movements through the process leakage the PCD. Okay, programmed cell death. The cause tissues are the overgrowth or shrink. Mainly are the what mainly are the part of the liver function. Okay, mainly livers are the some liver is the removed from the this type of the some are the unwanted materials. Mainly self removed self population increase the mass mark and loss. Mainly some are the cells also defensive mechanism by which is the damaged and potentially dangerous cells that can be eliminated to keep the individual healthy. Okay, the kind of the program cells, full variety of the cells, organisms, several distinct classes identified. We are them discussed with what means the cells having the no functioning. Okay, this is the PCD. The cells undergo the PCD might due to the some of the following reasons are the cell having no function. Cells have a no function. Okay, for example, mainly it is found in the chicks many cells of the early stage of the neutral differentiation. T cells are the so some are the especially in males and some many thymosis die before the mature into the T cells. And this T cells function is these are some of the cells are the apoptosis, the programmed cell. One are the cells having the no function. The second character is the death of the arm cell. Mainly are the during the molecular maturation of the T cell antigen receptors. The receptor recognize the own antigen, self antigens, and no the self they can identify the self antigen. Death of the arm cells. Okay, some of the potentially harmful are eliminated through the in this process, elimination process. Mainly are the related to the liver. Okay, mainly are the what are the following reasons? Some of the reasons of the PCD mainly cell having no function, death of the arm cells. Next one is the developmentally defective cells. Developmentally defective cells. A number of different kinds of the cells are the might have the arisen the defect damage at during the embryonic development. Mainly are the developmentally defective cells. Okay, developmentally defective cells. Next one is the elimination of the excess cells. Some of the mammals are the embryonic ganglia often to have the more neurons than required the enumerate the target muscles. For example, later excess of them not make the appropriate condition or the misfunctions will be eliminated. Mainly that the elimination of the excess cells. That are the characteristics of the one of the PCD reasons. The next one is the cells that have a finished their function. Cells that have a finished their function. Okay. See, this one is the after this is the development and differentiation. The next one, tissue homeostasis. They can form the cell division, the cell death. Cell death already PCD is also called as the cell death. Pathogenic cell accumulation. Next one, status. They can form the homeostasis. Next one is the pathogenic cell losses. Development and the differentiation. What is the development differentiation? The after is the independently rule of the development and differentiation, especially the embryo. The embryo it provides the switch off cells and the no longer need during the embryonal morphogenesis and the synaptogenesis. Next one is the immune system. What are the immune system? Mainly, the immune system adaptive programs are activated in the various situations. For example, elimination of the target cells. For example, viruses infected cells by the cytotoxic T lymphocytes. Okay, elimination mainly immune system. They can develop the immune system. Elimination of some of the autorecessive B and the next one T lymphocyte natural selection elimination is the by the thymus. 90%, 95% of the T cells have that migrate to the thymus are eliminated to the apoptosis. These are the mainly the thymus is one type of the gland is also eliminated. The cell damage, mainly cell damages. Okay, that the cell cycle already discussed the cell cycle. Mainly apoptotic program family protease named the capsosis. Capsosis. Okay, capsosis. What are the major pathways of the apoptosis? Mainly the intrinsic pathway. Okay, intrinsic pathway. Next one, extrinsic pathway outside, intrinsic interside, and the extracellular death ligands. They can form the extrinsic and intrinsic. Okay, next extracellular death ligands are the death receptor, mainly the activate the death receptors. Capsases mainly we discuss the capsases one, capsases 8, and then next one is the extrinsic pathway. The extrinsic pathway. Pathways of the apoptosis is the growth factor deprivation. Next one is the death receptors, DNA damage. Caspose is also death by the proteolysis. A family specialized protease named capsules is the central part of the apoptotic program. When capsules is one type of there are the specialized proteases named the capsules. Mainly the tumor suppressor protein is the P53 is the mainly the control the P53 has the both growth inhibiting and the properties is essential to the tumor suppressing activity. 50, P53 can be the separated mediated by the distinct pathways. P53. What are the main conclusions of the autopsy? The autopsies of the programmed cell death is the serious genetically controlled by the events. The removal of the unwanted cells. Autopsy is an important method of the cellular control and the any dispersion of the process leads to the abnormal growth is the cancer. It leads to cancer. Autopsy cells are the very characteristic morphology as well as the specific molecular feature. Induction and autopsy cancer cells are the mainly cancer cells are the malignant and benign cells also recognize the efficient strategy of the cancer chemotherapy, mainly autopsy cells also. When the induction of the autopsy, the cancer cells and the malignant tissues are recognized the efficient strategy for the cancer chemotherapy. Next points of the Software diagram marks cells and cells in chromatic connection, membrane labeling, and the nuclear collapse. So what is the analysis of the so Mitosis means death by the injury, autopsy death by the suicide. Person. Next slide. So, so, uh, yeah. Okay. The autopsis. Family of the protease is also called as the capsid. Capsid. Regulation of the autopsis. Regulation of the next one is the DNA. This is a very important. Next topic is the DNA structure and functions of the properties of the DNA. Okay. Already, nucleic acid they can divide the DNA and RNA, DNA and RNA. Uh, According to our syllabus, only DNA, RNA, DNA also is the both are. What are the main objectives of the DNA? Important pieces of the DNA. Chemistry of DNA, Watson-Crick model of DNA, replication of DNA, and next one is the transformation, conjugation, transduction of the in genetic recombination bacteria. Okay, these are the mainly DNA is the segment, especially gene is maybe defined the segment of DNA molecules. It is the basic unit of the heredity. Okay, the basic unit of the heredity. Mainly during the uh, okay, DNA is the polymer of the deoxyribonucleotides. RNA is the ribose nucleotides. The DNA is the deoxyribose nucleotides. The double stranded DNA. Mainly are the found in the chromosome and the mitochondria and chloroplast. Also DNA also mainly are the found in the chromosome and mitochondria and chloroplast. Mainly are the what are the main importance are the carriers of the genetic information. The heredity materials. They are the heredity materials. Okay, they are the heredity materials. Okay, what are the components of the nucleotide base, sugars, and phosphate is also called as the nucleotide base. The nitrogen bases, adenine and purines, the sugars, okay, phosphates. Already is the adenine and purines are also divided the bases. Purine and next one is the sugar, the phosphate, the main component of the DNA, the structure of the DNA. Mainly nucleoside. What is the nucleotide mean? Phosphate plus sugar plus base. There are also three base plus sugar plus phosphate is also called as the nucleotide. Nucleoside means sugar plus base is also called as the only nucleoside. Sugar plus base is also called as the nucleoside. Okay, these are the structure of the DNA and RNA. Sorry, DNA. Actually, nucleic acid they can divide the two types, RNA and the DNA. Basic structure of pyrimidine and purines. There are the pyrimidines. Okay, pyrimidines and purines. Uh, structure of the pyrimidines, purines, especially what are the pyrimidines? Cytosine, and next one is the thymine. Especially these are the both are the they can see the occurrence the mainly the available the content of the DNA, thymine and the cysteine also. Okay, uracil is the mainly they can see in the RNA. These are the pyrimidines. Pyrimidines. What are the CTU? CTU is the pyrimidines. Cysteine and the thymine and then uracil. Uracil especially they can see in RNAs. These are the pyrimidines. In the purines are the AG. Adenine and the guanine are the both are the purines. Okay, these are the nucleotide and nucleoside. Nucleotide means base plus sugar plus phosphate is the nucleotide. Only base plus sugar plus equal to nucleoside. Okay, nucleoside is the some of the nucleoside and the base analogs can be the used the anti-cancer and the antiviral drugs. Mainly the S 5 fluorouracil. Next one 6 merocarpurine is are the both are the anti-cancer agents. And next one is the azithromycin and the next one digoxin is the anti-retroviral agents are the both are the and next one is the structure of the DNA. Structure of the DNA is the main important features of the DNA is the mainly the single copy genetic information of the cell genome is a single copy of the cell. Gene segment of DNA is the molecule. Okay, what are the features? What are the important features of the DNA? Main is that the quantitative estimation of the DNA can varies the organism due to the DNA. The alleles are different forms of the same. Next one is the base components of DNA constant the organism various in the DNA is called deoxyribonucleic acid. The males are the containing the deoxyribose sugars and the phosphoric acid. DNA is the polynucleotide. DNA is the macromolecule. Okay, DNA is the hereditary substance. DNA is the life molecule. Okay, these are the DNA is the life molecule. Already chemistry of the DNA also. So chemistry of the DNA is mainly some of the proteins are linked with the DNA chromosomes. Uh, what type of the proteins are linked with some of the proteins? The histone protein is also histone proteins. The histone proteins are there contain the rich amino acid, lysine, in the arginine. Some are the net charges, the positive charges. All are the some are the especially some are the DNA can attach to some of the proteins. The histone protein. Okay, histone proteins. Already we discussed the nitrogen bases, the two types, the pyrimidines and purines. Okay, pyrimidines are the cytosine, thymine, and the uracil are the pyrimidines. Purines are the AG, mainly are the AG. According to Chargaff law, mainly A plus G is equal to G plus T is the A equal to T or G equal to C is the according to Chargaff law. Okay, mainly is the 3 prime and the 5 prime, and next one mainly is the 3 prime and the contains some of the which hydroxyl group. Okay, the traditional DNA sequence is the 5 to 3 strands, 5 prime and the 3 prime. Okay, mainly The secondary structure of the DNA, the anti-parallel polynucleotide chains around the some axis. Sugar phosphate chains wrap around the basis of the ATGC is the occupy the forming the complementary ANT. This is the Watson Crick base pair. DNA double layer cell together mainly is the form the hydrogen bonds, especially AT is the double hydrogen bonds. See, this one is the hydrogen bond. Okay, the hydrogen bond especially. What are the importance of the hydrogen bond? The molecularly attract to the electronegative atom, especially nitrogen, oxygen, the fluorescence atom. What are the hydrophobic nature? DNA is the one wall forces. Structure of forms of the DNA is the uh, early ADNA, BDNA, ZDNA. What are the difference between the ADNA, BDNA, and ZDNA? Property helix is the right side ADNA, right side BDNA, left side and ZDNA. How many base pairs are there? They contain the ADNA 11 base pairs. BDN is the 10 point approximately. It's also 11 base pair, 10 base pairs. And next one is the ZDN is the 12 base pairs. Okay. Next one is the mainly diameter of the ADN is the 2.5 nanometer to 5 nanometers. The BDN is 2 3.40 nanometers, and the ZDN is the 4.56 nanometers diameter. Mainly configuration of the glycosidic bonds with the ADNA anti-site and the BDNA anti-site. Next one, ZDN is the alternating anti-site. Next one, this is the normal hybrid DNA, B form DNA. The helical right-handed DNA is the BDN is the right-handed DNA. This is the right-handed DNA. Base almost is the the helix axis is the 3.4 angstrom. One turn is the helix is the 36 angstrom, 10 base pairs. Okay. Uh, the DNA double helical structure calls the round histone proteins. DNA bound the histone form. Nucleosomes, some of the fiber nucleosomes contain the approximately 146 nucleotides. Mainly, some of the histone proteins also. Mainly, histone proteins also attached to the histone core of nucleosome, and these are the linker. These are the histone core of nucleosome. Next one, linker of the DNA nucleosome. Nucleosome say the histone proteins are the histone proteins. Okay. Mainly, these are the Watson Crick model. We discussed the Watson Crick model. DNA replicate, mainly replication of DNA. Watson Crick model suggests that DNA replicate by the unwinding of the double helix. It shows the Matthew Melson, the Franklin Sethal is 1958. Using the technique is the DNA replication. Mainly, Nathal and the Melson, they can use for the Melson Sethal transport the growing heavy nitrogen isotope, 15 nitrogen isotope, and the medium contain only nitrogen isotope, the 14 and the 15. genetic recombination of bacteria. Uh, Genetic recombination of bacteria is the conjugation and transformation. Okay. Next one is the transduction. The three is the basic genetic recombination of in bacteria. What is the meaning of the transformation? Means the genetic process of bacteria. Mainly DNA from the donor is taken up the recipient cells. Mainly the mainly the donor DNA is the named the donor DNA is the only the portion of the donor genes are transferred to the recipient. Some especially the bacteria, especially the transformation is discovered in the Griffith. Uh, Transformation is the experiments is the Griffith in 1920, Everall et al. 1944 also. Mainly is that the extracellular DNA is brought into the cell. One of the strands hydrolyzed by the DNA degrading enzyme. What are the degraded DNA degrading enzymes? The single strand part in the cell incorporated the host genome from the crossing over. Conjugation that the developed by the Ledenberg and the Edward thought of discovered in the E. coli. Okay, the conjugation E. coli. Next one is the transduction is the they can develop the Ledenberg 1952. Will Salmonella, Salmonella typhi. Okay, mainly native which is the identified the India native different, especially 19 GB watts model of the DNA mainly the found in the protein mitochondria of the structure. There are the DNA, DNA structure is the files of the histone H1 B1, the different types of histone protein. DNA on histone forms of the nucleosome is genetic information. Differentiation development in therapy to charge the A plus equal to T plus the according to charge kapla 1 plus 1 A plus equal to T plus the charge kapla what the double the 20 amps का very the 20 amps का 20 amps का next is the main the मतलब is the 20 amps का next is the thermal is the potassium is the 34 amps का each the one pair of the 3.4 amps का the pair point is the 3.4 structure the structure of the structure of the B form of the part is the example of the Watson Crick model of the B form B the left hand is the A, the right hand and that is the left hand of the the small mitochondria some things also mitochondria approximately 70,000 within the base pair. Mitochondria 37 the mitochondria is DNA also mainly mitochondria is mitochondria there is the cell structure is the cellular growth type the mitochondria and the DNA is the nucleus genome size approximately 70,000 mitochondria. Next, nucleus part is chromosomes, different base pair, that is the subclass of the light and the growth factor. Uh, the, the so palindromic mean, especially, for example, the Nitin, the DNA duplex process the sequence of the some of the opposites have two ends span. Some sequences decaying the restriction endonuclease using the genetic engineering. For example, the similar to palindrome having the words the both the forward and the backward direction. For example, Nitin. So these are the also Nitin. Malayalam. What is the meaning of the palindromic mean DNA mean? Similar palindrome, especially the both the forward and the backward direction is the same. Back, backward direction and the forward direction. When the inverted repeat occurs within the each individual strand of DNA, the sequence is called the mirror repeat palindrome DNA. Palindrome sequence can from the alternate structure with the started pairing triplex DNA standard triplex and standard now with the forest or the cost of the PCR in a with that uh, structure and DNA according to syllabus, bacteria, transcription, next one, conjugation, transformation. These are the types of the DNA, and next one is the cell absorption. Next one, cell cycle. Uh, 6 is over. Next one is like 5 also is over. But it's not annual examination point of view. What is the mechanism of the autopsy? Describe the autopsy, the definition of the autopsy. Next one is the DNA structure of the DNA, importance of the chemistry of DNA. Next one is the Watson shift model of DNA. We'll discuss the Watson shift model of DNA. Okay, write the account of the DNA synthesis, the chemical nature of the DNA. These are the very important. Okay, this is the DNA and cell autopsis. Next one.